So today's webinar is about using flow chemistry in pharmaceutical research and development, particularly the early processes of drug discovery. I'm sure lots of you have heard lots of things about flow chemistry and the various advantages that it can have over traditional batch processes. Maybe even you started using it in your own research, but it has advantages, particularly in certain kinds of reactions, but also more broadly. During this webinar, we'll be looking at some of those benefits in a bit more detail, especially using modular flow reactor setups that have the flexibility that you might need in that kind of discovery and hit to lead environment, and then finishing with an example of a company that is putting these things into practice as it refines those screening hits into lead compounds that are going to progress further through the drug discovery process. We've worked with Syrris today to bring you the information in this webinar. We think that you're going to be interested in it, and we've got some really high-level experts.
So a brief introduction to Cirrus. So we're a company that was founded, well, over 20 years ago now, to look at a specific application, looking at chemistry using microfluidic devices, what we now know as flow chemistry. We are the longest established flow chemistry company, having released the first commercial flow chemistry apparatus all the way back in 2003, but we're the only company that addresses both batch and flow markets, which gives us an excellent position to help our customers. So as our years denote, we have instruments in all of the top pharmaceutical companies, in CROs such as Vinalis, and then a high number of academic institutes. And just to give you an idea where we are, because I'm sure this is a global audience, we're based in a place called Royston, which is just outside Cambridge in the UK. The pharmaceutical industry continues to go through changes, both in the way that it approaches drug discovery, but also in the way that it uses and develops new enabling technologies. And if you look at the two infographics here, you can see that the majority of the cost is in that early drug discovery process. And certainly in that early hit to lead and lead optimization processes, we need the greatest number of chemical entities. So one thing that is certain is that we need to speed up that process, bring the cost down, and find a more effective way of making lots of active compounds that are generally novel or structurally diverse in increasing numbers in shorter timelines. So these are our real drivers. But in the drug discovery process, in that early process, the development of new drug candidates involves this iterative learning cycle to identify those new clinical candidates, especially in hit and lead finding phases. This learning process is critical in identifying potential new drug candidates. And if we can speed up this decision-making process, it's certainly evident that we become more efficient in our drug discovery. The faster that we can go through these learning cycles, these iterative cycles, the faster we can get to our lead compounds. Now, there's been many approaches to the way we do this over the years, going from early stages where we were using that knowledge, intuition-based approach to design compounds, where we'd make them singularly on gram scales and then feed them into animal models. You know, that probably wasn't the most efficient method, but probably one of the most striking kind of technological developments over the years has been the ability for us to screen compounds in vitro in increasing numbers. And it's that high throughput screening that's led to reducing the bottlenecks in this drug discovery cycle and the need to produce and have methods for high throughput chemistry to work alongside high throughput screening methodologies. And we've developed these over the years through parallelization, so parallel chemistry, combinatorial type approaches to make libraries of compounds in increasing numbers. Now, newer technologies are continuously being developed, flow chemistry being one of those, and it's been increasingly adopted in this early drug discovery process. And this is what we'll be talking about. So before I go further, I know many of you will be familiar with the drug discovery process, but not all of you may be familiar with flow chemistry. So I want to give a brief introduction to flow chemistry. And I always start these presentations off when I'm talking about flow chemistry to say that flow chemistry is just a synthetic technique. It's complementary to traditional batch methods. We use the right synthetic technique for the right chemistry and applications we want to carry out. But fundamentally, flow chemistry is a tool that allows us to control our key reaction parameters, whether that's reaction time, temperature, molar ratios, mixing or additions. So we all know how to carry out a batch reaction. You know, this is our training, this is our bread and butter. We've been using the same technology for hundreds of years. Flow chemistry is not changing it's not changing the chemistry that we carry it out. It's just using a different reactor technology to continuously produce our reactions. So it's a slightly different way of carrying out chemistry.
Now, timing of multiple reactions in a slug flow system would be a daunting prospect for any manual system. This, however, can be elegantly controlled by the software in an automated system. Now, note here the timing of collections is pre-calculated and controlled by the software. And here the individual reaction slugs are all timed from the point of entering the injection loop. We don't require each reaction to clear the entire system before the next can enter. And this is an appreciable time saving when we consider this effective stacking of reactions. Now remember, all the reactions are running through an identical continuous process, so all are under the identical reaction conditions.